This is the 11th episode of Lab Rat Chat, a podcast dedicated to informing the public on the importance of animal research and why it is 100% necessary for continuing to make medical advancements for both human and veterinary medicine. This podcast is supported by Americans for Medical Progress through the Michael D. Hare Fellowship, which is awarded annually to support projects that inform and educate the public about the critical role of animal research in furthering medical progress. The fellowship honors the late Dr. Michael Hare, a renowned board-certified laboratory animal veterinarian who dedicated his career to scientific and medical advancements, and he was deeply committed to animal welfare and advocacy. In this episode, Danielle and I talked to Dr. Dale Cooper, a board-certified laboratory animal veterinarian, about some of the many common misconceptions you hear talked about when it comes to animal research and pharmaceutical drug development testing. So sit back, relax, and enjoy this 11th episode of Lab Rat Chat. Welcome, everyone, to the 11th episode of Lab Rat Chat. It's been a little while since our last episode, so I'm excited to get back on the mic with Danielle and our guest today, Dr. Dale Cooper. He's a laboratory animal veterinarian. But before I introduce him, I just want to remind everybody to please follow us on social media. On Twitter, we're at the Lab Rat Chat. Follow us on Instagram, our Facebook page. You can Google it all. You can find us there. Leave comments, tell your friends, family about it, and let's keep growing our subscribers for the show so we can keep the show rolling for the foreseeable future. And that being said, I just want to jump right into today's episode. We're going to talk about some misconceptions surrounding the world of animal research, and we have a great expert in the field to talk to us about this today with Dr. Dale Cooper, a laboratory animal veterinarian. Welcome to the show, Dale.
Hello, thank you. Glad to be here.
Yeah, thanks for coming on. We really appreciate your time. And if you could you just tell our listeners and us a little bit about your background, what made you interested in veterinary medicine and laboratory animal medicine specifically, and just give us a breakdown of your journey to where you're at in your career today?
Sure. I liked animals growing up, and I think that's kind of the prerequisite for anyone who goes into veterinary medicine. And I lived in a small enough area that I didn't really know what other professions were out there that allowed you to deal with animals. So the veterinarian route seemed the right one to me. And I majored in wildlife ecology as an undergrad. And at that point, started getting interested in the whole process of research. I realized that, there's a lot you can do to work with animals that don't just involve the medical care. I was on my way to vet school. I got in, went through, loved it. I worked for about 3 years in private veterinary practice. And, we just was starting to realize it really wasn't exactly what I'd been looking for. One of the things I think that helped direct me towards laboratory animal medicine is at the time I was working in mixed animal practice. So I did both small and large animal. And I realized I actually really enjoyed the large animal medicine. Herd health, there's a big emphasis on preventative medicine. And the clients that you worked with were almost like a partner. And you really were working side by side with them rather than being sort of in this, maybe more authoritarian relationship that you have between a doctor and a patient or a client in maybe companion animal medicine or really like human medicine. And so I really enjoyed that interaction a lot. But time I was living in Wisconsin and I realized, A, I don't really want to be doing surgery on a hay bale, and B, I don't really want to be working outside in the middle of winter. So
yeah,
because I'd had this prior experience with research in my undergrad, I started looking into what other options there were and ended up doing a residency in laboratory animal medicine and becoming board certified by the American College of Lab Animal Medicine. And so I've worked in that since. I've worked for academia, I've worked in a pharmaceutical company, and I'm in contract research now.
Awesome. Thanks for that breakdown. It's funny, so many of the lab animal vets that we've talked to on the show, they start off in another, within veterinary medicine, within another field of veterinary medicine, and have always, obviously transitioned into the field. I think we've had one so far that actually, after vet school, pursued a residency and pursued lab animal veterinarian career right off the get-go. This is very common in this field, it seems like.
Right. I mean, you're seeing more and more where people go directly into it. And a lot of that has been, honestly, because of things like this, where people are talking about it more, talking about it as a viable career alternative and all the advantages and just how incredibly interesting it is.
Like you said, it's more of a recognition of this field. I think some veterinarians going through school don't realize that this is really a career option. I mean, I have classmates still that don't really know what lab animal medicine is. I had one student, before listening to this podcast, one classmate, she said, I think I may have said this on the show before, but she said she thought the world of animal research and being a veterinarian within that field just meant you just euthanized animals all day. And she listened to this podcast and completely changed her perspective and is pursuing externships and potentially a career into this field. And someone who had never thought about doing that before.
Well, I think even outside the vet career, even just for other people who just love animals, because, I'm not a vet, but I did the animal science degree and I was like you, I did not want to work outside in the winter. I kind of wanted a temperature controlled 9 to 5 style job. And I found lab animal research junior year of college, and it's just become the best career.
Oh, yeah. There's just so many different opportunities, so many things that people can do, so many paths your career can take.
Yeah, and most of the vet techs I've come across with in this field also worked within small animal or mixed animal practices before switching as well. So it's common, not just for the veterinarians, you're right. I mean, across the board. Well, let's get into the meat of the show really today, what we want to talk about, which is the misconceptions surrounding this field. And you wrote a pretty fascinating article a few years ago now for Speaking of Research, and I'll provide the link in the show notes for everyone out there. If you want to go read it, I recommend that you do. It's called The Predictability and Utility of Animal Models. And it broke down and explained some of those common misconceptions we hear about, such as like 9 out of 10 drugs shown to be effective in animals will fail in human clinical trials, or that a very tiny fraction of drugs, like 0.0002% tested in animals ever end up attaining FDA approval, or that, flip a coin and that's about as lucky as you'll get in like predicting drug utility in humans from animals. So in order to address these misconceptions, I think we first should start by breaking down some of the phases of the drug development process and maybe talk about the beginning phases, which include things like in vitro methods and cell cultures grown in petri dishes and computer modeling and that kind of thing.
Yeah, so that's definitely one of the big misconceptions. And there's kind of 2 facets to it. Some people will think about drug development and in particular animal research as if you run one study on one drug and that gives you all your answers. And that couldn't be further from the truth. It's a process. That process starts, honestly, even before in vitro testing, where the chemists are designing the drug. You initially start with looking at what the market need is, what are some unmedical needs, what's the hypothesis as to how you might treat a certain disease. They're already delving into databases and chemical libraries to see, what's known about molecules that might be useful as drugs. How do they interact with different parts of the body? Can they impact the physiologic mechanisms that lead to some of these diseases? And so there are computer programs now that essentially are designing and screening out drugs simultaneously that are incredibly powerful for going through, large numbers of drugs. And from that, you essentially generate what's called like a library of drugs that might be against a very specific target or disease mechanism. And it's at that point that you would start doing the in vitro testing, looking at, how these drugs interact with cells in culture. What receptors do they bind to? What do they do when they bind to the receptors? Do they activate something? Do they deactivate something? Are they toxic? What types of genes are being expressed? Can you alter gene expression? As a veterinarian, this is not my area of expertise, but there are hundreds and hundreds of different screening assays that are used, and you're constantly narrowing down that pool. And so for every drug that gets on the market, the pharmaceutical industry estimates 10,000 plus drugs have been screened out along the way. And really 90% of the candidate drugs never make it out of the laboratory. Between the chemistry, design, and screening process and the in vitro screening process, that's eliminating a large number of these candidate drugs from testing pool. And it's only after you have gone through those stages that you would start going into animal models. And the earliest stage of animal models is what's sort of like a proof of concept, or sometimes it's called efficacy testing. But you're really looking at, okay, that it does what it's supposed to do in a cell culture, but cell culture is a very simple system. And even some of the newer cell culture things, such as what they call organ on a chip, where they may actually have several different cell types growing in specific compartments, but they have communications between them to simulate like blood supply and that sort of thing. But those are very simplistic models. You get into a living body and there's a lot going on. And frankly, there's a lot going on that we don't even understand. You have to see kind of what happens in a real life situation. And again, you're narrowing down things from that point. You have a smaller candidate pool that you would then take into safety testing. And the safety testing, you're really looking for, what are the potential side effects? What dose do they occur at? Are they reversible? If you're a clinician and you're giving a drug to someone and you see a side effect, obviously one of the things that's going to be important to you is, can you make it go away if you stop dosing? Can you treat it? So there's all these steps that things go through, and that's before they would even start doing trials in humans.
So that kind of breaks down my next question, which was, once you've done all that screening and you have the drug candidate identified, based on your description, it's really not an option to just go straight into testing that drug in humans because you do want to test safety and efficacy.
Right. It's about risk tolerance. So the data that exists right now suggests that if you only ever did in vitro screening using the tools that we currently have, you would only predict about 30% of the potential toxicities in humans. And so what that would mean is you come in, want to sign up for a clinical trial, and I'm like, okay, you have a 70% risk of not only having something happen, but we don't know what it is. So we don't even know how to monitor for it. And it just might kill you.
Yeah, right.
So you have to ask, are you really willing to do that? And, of course, the answer is no. It's already hard to get people to sign up for clinical trials. And we have largely de-risked them through this iterative process that involves in vitro testing as well as animal modeling. And imagine if you were to try to recruit people to do this without all of that information.
Yeah, I wouldn't be signing up. I wouldn't sign up as it is more than likely. But without all that information, it'd be really tough to get me to sign up. And I'm just speaking for myself. I can't speak for other people, but like you said. And I think, severe adverse effects in the beginning phases of human clinical trials are extremely rare. And I think we have to thank use of animals for that.
Correct. And generally, they know what to monitor for because they know what things have been seen in animals. And so you can detect issues in human clinical trials relatively early before they become severe because you have that information from the prior work.
Yeah. So that's basically what these researchers are looking for then in the animal model before they're able to get approval, before they move it into human clinical trials. It's just looking for efficacy, lack of toxicity and that kind of thing.
Yeah. And interestingly, there's a little bit less emphasis on efficacy because, ultimately your efficacy model is your human. So that's where the phase 2 and phase 3 clinical trials come in. We know there are a variety of differences in how any species responds to disease. And so animal models, interestingly, tend to be less effective at efficacy modeling than they do at safety modeling. But what you can develop is proof of concept. You can say, look, we know that this particular gene or receptor or physiologic process is involved with the disease. We can measure if that is being impacted during the animal studies and, believe that will translate into humans. And there are ways of kind of even verifying this ahead of time. They'll take human tissues, liver, that sort of thing where a lot of drugs are metabolized. And you can actually look at how things are interacting with human cells. Even though it's not the complete system, you get a pretty good idea of, is this animal model likely to be predictive of what's going to happen in humans? It doesn't have to replicate the disease completely. It just has to show that it has some of the same mechanisms that are present in humans. And so that's a big part of it. But then with the safety testing, you're looking for a variety of things. You're looking for what signs you might see, what drug dose level you might see them at. I mentioned before reversibility. If you stop dosing, do they go away? Can you treat them? And then ultimately, you're looking at margin of safety. So you have an idea of what the effective dose is likely to be because of this early efficacy testing and some of the in vitro and computer modeling that's done. You know where you're likely to see toxicity. Typically, you will not proceed with a drug until you have anywhere from a 10 to 100x safety margin, because it's very likely that once you get into humans, you're going to have to adjust that dose in order to achieve the efficacy that you want. And so if the safety margin's too narrow and your efficacy dose overlaps with your toxic dose, that's not a useful drug. Yeah, there's a whole series of things. And then it also gets into a lot of detail. They may look for specific effects on fetuses through the use of, rabbits and rodents that have really high reproductive capacity. You can actually generate a lot of data to see how the fetus is affected. We do what's called safety pharmacology, where you're looking at the effect of drugs, not at a toxic dose, but at a more normal clinical dose. And you can pick up subtle effects on the heart, respiration, central nervous system function. So there's, again, just a whole series of those studies that go into this.
I was just going to say, a lot of that work then with in utero and babies and, pregnant animals is being done in animals then, right? Because I imagine you're not getting pregnant women to sign up for human clinical trials that are willing to test the effects on their babies.
FDA won't allow it. And one of the things that I think is also a big misconception is people assume that animals take on all the risk associated with drug development. And in fact, it's very much a shared risk between animals and humans. So the way the safety guidelines read, you have to show safety in an animal model, actually in 2 different animal models, for the amount of time that you're likely to then dose the drug in the human clinical trials. And so an initial human clinical trial is only a single dose. And so you have to have single dose plus like up to 1 month repeat dosing in animals in order to be able to get approval for that single dose. Well, the same thing happens before you can dose women of childbearing age. You have to have done the reproductive safety studies in animals. And so it's very much a planned process, and it's not the same map for every drug. They really do look at what's the target population, what's the medical need, and they plan the animal studies so that you don't get too far down that path before you say, you know what, this actually isn't working. We need to stop.
I was just wondering, because my wife and I, we have 4 kids, and whenever she's been prescribed something for nausea, like morning sickness or heartburn, or wants to take medicine for migraine or something like that, and you go and look up the drug, everything's like, I don't want to get into different categories, but it's always, usually, it's unknown how this will affect the baby. Just consult with your physician.
Yeah. So a question I had, we're talking about the use of this in, mice and rabbits and everything. Can you just test a drug candidate in one species? Do you have to try different species? Are there certain models that are better that would more closely mirror a human? What's sort of the process with animal model?
Every animals are a model. And by definition, a model is not an exact replica of, whatever it is you're looking at. All animal models have strengths and weaknesses. Some are more effective at predicting some types of toxicity or efficacy than others. And so for that reason, you do actually look at everything in more than one model. So FDA specifically requires a rodent and a non-rodent species. And that actually goes way back to the 1960s when thalidomide was approved in Europe. And it turned out it caused birth defects that were not detected in rats, but could be detected in different species. Well, at that time, rodents were the only model that was required. And since then, there have been studies that have shown that by increasing the number of species that you test in, you significantly improve your ability to detect toxicities. But we definitely know that, there's certain models that are better for some diseases than others. So the pig model is one of the better ones for looking at how things are absorbed and tolerated by the skin, because they have this relatively low hair skin that's thick, kind of like humans do. Cardiovascular function is actually very similar in pigs to humans. But for other things, for immunology, a monkey might be a better model. For a lot of just general drug metabolism thing, dogs are a better model. The scientists have to look at each of these things. And again, they talk with FDA as part of the development process. And FDA has, access to all of the drug testing that's ever been done. And they can say, look, can't tell you what your competitors are doing, but we can tell you in our experience, this is the model you want to use for this type of drug. Yeah, it's very much a planned process.
Could you tell us a little bit about, why is it that so many of these drugs, once they get into human clinical trials, are failing? If the animals are able, I guess they're not completely able to predict which drugs are acceptable for human trials and which ones aren't. Is it just lack of efficacy? Because I guess that's what you were pointing out earlier. We're looking more at safety margins.
Lack of efficacy is one reason drugs fail in clinical trials. Sometimes, they just don't work as well as they thought they would. And so a drug company may say, well, you know what? There are already drugs on the market that are as good as this. Let's not pursue it. What we really wanted was a drug that was better. But there are safety issues as well. So animal models are pretty good at detecting if there's toxicity in the animal, whether or not it's likely to occur in humans. And so they're actually very useful for screening out those 10,000 drugs that you have to filter through before you get to that one effective one. That doesn't necessarily mean, though, just because it was safe in the animal model, that it's also going to be safe in humans. And there's a lot of factors that go into that. Some of it are just the physiologic and genetic differences. But the other thing is the human population is a very uncontrolled research model, if you will. So when we do this testing in animals, we are using, genetically defined animals with defined health status living under controlled conditions. You get something into the human population and we just don't have that level of control. And so there could be environmental factors, concurrent drugs that they may be taking, whether legal or illegal. And all of those things can impact drug safety in the real world. So you have to look at the positive predictive value is one of the terms that's used of safety testing. It actually tends to be pretty high. The negative predictive value, meaning, it was negative in an animal, therefore it's going to be negative in a human. That's not as high. That's where I think that 50% flip of the coin statistic might come from, is the negative predictive value is definitely significantly lower. But that's true for in vitro testing. So if you say, well, let's just do in vitro testing as a replacement for the animal model. No, the in vitro test will tell you if that drug is toxic. But if it passes the in vitro test, there's still a relatively high likelihood it could still be toxic in subsequent animal studies or human studies.
So say a drug makes it all the way through clinical trials and gets approved for marketing. You see those commercials for a new drug, and then you have that guy at the end who talks really fast and goes over the side effects, may cause headache, nausea, or possibly death. And he says it really quickly. How can a drug or a treatment plan has to have that little claim at the end, I don't want to die from trying something new. How does that make it all the way to the market?
Right. So again, it's about margin of safety and using the drug for the proper indication at the proper dosage. When they report things in the package prescribing information, which is essentially what they're talking about is what's in the drug package insert, FDA requires that you disclose everything that happened during both your animal studies and your clinical trials. People may die during a clinical trial for reasons that have nothing to do with a drug, but you don't always know that, right? I mean, your clinical trial may involve 1,000, may involve 10,000 patients. Well, that's a lot. When it gets on the market, you have millions of people. And so you don't necessarily know just because something only happened in one patient that it's not going to turn out to be 10 or 100 or something when it gets out into a larger population. But FDA has to err on the conservative side, make all this information available. So doctors are aware of it, patients are aware of it. And it's just the requirement they have is that you rattle off all that stuff when you advertise it on TV. And it's so people don't be like, it's fine, no problem. I'll just go ahead and take this. No, drugs are serious business. You really should only be taking them when you have a legitimate medical problem. And that's why they require, a physician to prescribe it, because there's always the potential for side effects. One of the, I guess they consider him the founder of modern toxicology, says it is the dose that makes the poison. One of the examples that's used is you can die from too much water, not drowning, but drinking too much water can kill you. Well, we generally think of water as a good thing.
You can have too much of a good thing, that's for sure.
Yes. So people have to respect that.
Yeah, I always love it when a side effect of a drug is what it's trying to cure. You have side effect, you're trying to get rid of your migraine, and the side effect is migraines.
Yeah, is that a side effect or is that just the drug not working?
Yeah, who knows, right? Does it increase your migraine? Now you have a double migraine.
Physiology is very complex. Everything exists in a balance. And when you start changing that balance, sometimes compensatory mechanisms come into play. There's individual variability. You have 2 copies of every gene and how many alleles for each of those. And so it may be that there's a mechanism that is valid in one patient, but not in another. And this is really what the drug companies and FDA are seeing as the future of medicine, is personalized medicine. Once we start understanding these genetic factors and epigenetic factors that contribute to disease, it may not be a matter of, here's one drug that treats this disease. It may be this is the drug that treats your disease. You can imagine some of the complexities with going that route and the costs involved with going that route. But our current understanding of disease processes suggests that that's the way it's going to be in the future.
Yeah, and that might be better in the long run, and that's probably a topic for a whole other episode, because we could spend a lot of time about personalized medicine and epigenetics and all that stuff. So in the end, it seems like this road to FDA-approved drugs is long, it's drawn out, it's an expensive process that has a very low success rate, basically. It's not necessarily the animals. What should people take away from all of this when they read the headlines in the news that animals aren't effective because 9 out of 10 drugs fail to make it through human clinical trials? Is it right away? Because what they're aiming at is right away we should argue that animals are a useless model and we shouldn't be using them. But I think you disagree. I disagree. But I don't want to tell you your opinions based upon what we've talked about.
To some extent, I kind of flip the question around. If you know of a better way to do this, the market is wide open for you because it's long, it's time consuming, it's expensive, that sort of thing. If you can find a less expensive way to develop a drug, you're going to make a lot of money. And the pharmaceutical companies themselves will probably pay you for that ability. And so, not to be, I am being catty about it. It's one thing for the animal activists to sit there and snipe from the sidelines, but are they actually doing anything useful to try to change what's going on here? Are they funding research into alternatives? Are they getting directly engaged in the drug development process and trying to come up with other ways of doing things? And I think we know what the answer to that is. This is where we're at. This is where we're at in terms of our current technology, our current knowledge. I think that's really what people have to think about is, what are the alternatives? Are there better ways of doing this? And I think we've done ourselves a disservice in the pharmaceutical business by responding to the animal activists using sort of extremist language. So they're like, you need to use alternatives to animals. And our answer is there are no alternatives. Well, that's actually patently untrue. As I already talked about, there's a lot of alternatives and we use them all the time. A high percentage of the work and the drug development work that's done and the screening of test articles is done using non-animal alternatives. You reach a point where those don't tell you the remaining answers. You use animals at that point. And so I think we need to maybe change the language a little bit. Can we improve some of these alternative models? Can we, validate additional approaches to drug development? And the answer is yes, we're doing it all the time. Every time a drug gets developed, the pharmaceutical companies will put this information back into their databases. And the next time they're developing a drug, they're going to say, well, last time we made a molecule that looked like this, this is what happened.
Do you see any in the future, the near future, or even distant future, an actual replacement for animal models? Based on everything we've talked about today, it doesn't seem like that's really feasible at this point.
I don't think so. I know, a lot of people talk about it. Even FDA has been pushing this. I think what you'll see is a continued refinement and maybe a decreased dependency on animal models. But almost by definition, in order to model something exactly, you need to know everything about it. Well, by the time you know everything about it, there was no longer any problems to solve. 100 years from now, we will probably use animals differently than we do now, but I suspect we'll still be using them. 1,000 years from now, I don't know how long it'll take. It is not in the near future. I think back to just my career. And, I remember when we started out, they were just starting the whole human genome sequencing project. And everyone was so excited about this. We're going to discover all the genes. We're going to have the sequence to everything. We're going to be able to just design these medicines that are very specific and we'll know everything. They got all that data. They started doing a lot of transgenic models, knock-ins, knockouts. And it was interesting because, again, these models didn't always work out the way they were supposed to. And people started realizing, well, geez, there's actually more than one gene that affects this. And there's compensatory mechanisms and there's all kinds of things going on. And so now we're sitting here looking at this library of, 20,000 some genes, not to mention all of the intervening DNA that we're starting to learn does some stuff too. And we're like, oh my God, there's a lot that we didn't know about. Of course, we're learning so much about epigenetics. Microbiome has become very much a hot topic. And that's where, the environmental bacteria and other organisms end up having direct effects on your physiology. And we probably shouldn't be surprised by that, right? I mean, you look at a human or an animal and it's pretty fascinating that it works at all, right?
Yeah.
So, but here we are walking around, talking to each other, that sort of thing. And by all accounts, we started out as a mixture of molecules, Do I envision it where it's never going to be that way? No, I really don't. I'd like to be proven wrong. I'd like to think of a world where we know everything, but I don't know. Maybe that wouldn't be fun either.
Yeah. Yeah. I mean, it's almost unfathomable to think about if you think about it on the cellular level and all, the hormones and receptor binding sites and all of that stuff that we would ever understand completely how each and every single little cell or hormone or gene or whatever could work in the body. And that's just one person, like you handed out earlier. Every single person's going to be different also.
Right. There's no question we're making enormous strides. There's exponential growth in our knowledge, and there's exponential growth in computer technology that allows us to better analyze all of this. I don't know. It's a big job. I think we oversimplify what we're up against.
Yeah. And I mean, I think we would all love to see the day that we actually had a viable alternative that predicted safety, toxicity, efficacy, everything that we needed without using an animal model.
The people I work with love animals. They're not using them because they're callous or uncaring. I mean, they really value them. They come to me all the time. What can I do to improve these experiments? I think we do very well in terms of treating animals humanely, but when you're studying disease and you're looking for side effects, it's not a perfect situation. And so the people really care a lot, though. that's what keeps us working at this. It's better scientifically, it's better, frankly, economically, if you can either improve animal models so that there aren't any side effects or anything, or to use non-animal models. But it's also something people care a lot about ethically. The people that work in the business. We don't need people from the outside telling us that we need to care. We already care.
So we've been asking this last question to a lot of our guests, or all of our guests. We asked them how to describe what they say to people when you're asked about what you do for a living. And I'm guessing I'm going to know how you're going to respond based upon your responses to some of these questions today. You bring up that you work with animals and it's animal research, and, what are their reactions, and what do you say to them when you receive, that look like that?
I do talk about it. Admit I'm somewhat selective. If you're in an airport and you don't really want to get into a long conversation with someone, you may kind of brush it off. But if I have the time and I'm willing to talk, I tell them what I do. Of course, as a veterinarian, I have a little bit easier of a job because I can say, hey, my job is taking care of the animals that are used in research. But, if you're a scientist, you can also talk about the work that you're doing, the people that you've impacted. I know our company actively provides feedback to employees on drugs that we have worked on that have gone onto the market, and getting patient testimonials and just seeing what the real life impact is of making a difference for people. We talk about the fact that animal research benefits animals. All of the veterinary drugs that are available came through the same exact process. And so we also talk a lot about just all the care that's put into it, talk about alternatives. And what I have found is that the vast majority of people, once they understand that there are a lot of controls in place and that this isn't just a willy-nilly thing, feel very differently about it. And frankly, that's my target audience. I don't have any illusions that I'm going to take a hardcore animal activist and make them think differently about this. But I really care a lot about what my mom thinks.
Yeah.
I can't explain it to her. I figure maybe that's not a good thing. I care about what my family and friends think. And so I care about what I think. I have to be comfortable with what I'm doing. I think I'm an ethical person. I kind of just talk through how I view this myself and why I think it's morally acceptable to do this. And of course, why it's scientifically necessary too.
I think that kind of wraps up all the questions we had for you today. Is there any final thoughts that maybe we didn't cover that you want to kind of bring into this episode, or things we didn't?
I kind of alluded to this before, but I talked about risk tolerance. Ultimately, we always say, well, you have to use animals. No, you don't. It's a choice. As a society, we've chosen that this is what we want to do in order to treat disease and that sort of thing. I respect that some people may not view it the same way, but what they have to understand is they can't make a choice that takes my choice away. If someone personally is against using animals in research, they can stop going to the doctor. They can stop driving consumer demand for drugs. They can not threaten to sue someone if the drug is toxic because it wasn't tested in animals and no one knew that it was toxic. There's a lot of things that people can do as individuals to take control over their own ethical beliefs. But I also have the right to make a choice, as does everyone. And the reason that the process exists is we're in a democratic society, and the vast majority of people, even if they won't admit that they're okay with animal research, they are driving the demand that essentially, is there, asking us to develop drugs, develop within a certain time frame, within a certain cost, and with a certain level of safety. And so I think that's, again, people just have to really ask themselves, if I really believe this, what am I willing to give up? It's very easy if you're, an 18 to 22-year-old college student to say, well, I don't really care about all these diseases because you've never had one. You may not even know anyone that's had one. But you ask the same people that have family, people they care about or their own diseases, they feel a little bit different. I just think maybe people need to take a real hard look at their own motives when they get involved in some of these discussions. And I think by all means, we should be having active, intelligent discussions about ethical issues. I don't think we should be threatening each other, committing violence, that sort of thing. And that's unfortunately what sometimes happens. I don't even think you should be nasty about it. You can have a respectful conversation with people and disagree.
Yeah. Disagreements we have to have disagreements as a society. We all agreed that, what kind of society would that be? It would be a bunch of robots. It would be just robots walking around. Disagreements are normal. It's about having honest and open conversation. So you said that very well. And I appreciate your insights, and I appreciate you coming on the show today and giving our listeners and giving Danielle and I even loads of new information and, looking at things from a different perspective that I hadn't even really thought about before with some of your answers to these questions. So again, just thank you so much for coming on today. And for all of our listeners out there, I'll just repeat the fact that please follow us on social media, Twitter at the Lab Rat Chat. We're on Facebook. We're on Instagram. You can email us if you have suggestions for what we should talk about. If you want to be on the show, if you want to suggest a guest that we should talk to, email us at labratchat@gmail.com. And thank you, everybody, for listening. And we'll be back with another episode soon.
Thank you.
Well, thank you, Jeff. Thank you, Danielle.